the glucose is normally an uncharged molecule. So it cannot, and it's a very big also. But it cannot enter just like that into a cell. It requires specific transporters. Transporters we already studied yesterday. So once the glucose enters, like this door, through this door you have entered into class, and through this door only you can go out. If I spend more time looking at the board, then few of you will go out. That's why I don't look at the board. Isn't it? So same transporter can be used for glucose to outside. So how to keep them inside? How to keep them inside? Glucose being uncharged, they can enter and go out easily by the same transporter. So as soon as the glucose enters, a reaction takes place which require very small amount of glucose. That means what? Very low km, except few tissues. Most of the tissues contain an enzyme which is having very low km for glucose. That means small amount of concentration of glucose will trigger the reaction and glucose will be converted to glucose 6-phosphate. Now, by adding phosphate, what changes we bring about with the glucose molecule? Yes, the glucose becomes charged, phosphate is negatively charged. So now the glucose being charged molecule, it cannot escape the cell. It is trapped. Right? So the other enzymes of metabolic pathway can participate and can utilize glucose 6-phosphate as well. So the first step of the reaction of the metabolic pathway is very, very important. Such reactions which play a crucial role for any metabolic pathway like hexokinase did in this pathway are called dedicated step. Once the glucose is converted to glucose 6-phosphate, then it cannot go out. So it has many ways to go. It can go to glycolysis, it can go to glycogen formation, it can go to formation of gluconic acid, etc., many ways. So for every pathway, there is a first dedicated step. For glycolysis, it is catalyzed by hexokinase, except few tissues like liver, where instead of hexokinase we have glucokinase, which has, on the contrary to the hexokinase, which is very high km for glucose. Name is glucokinase. It is more specific towards glucose, but it requires more amount of glucose to get activated. Why? Because the liver uses glucose for storage purpose. So storage is possible when we have excess. So that is why liver function on the glucose molecule when the blood is having plenty of glucose, excess of glucose, unlike other tissues. Fine. So the glycolysis pathway, can be of two types, aerobic and anaerobic. The aerobic glycolytic pathway, end product is pyruvate. Anaerobic glycolytic pathway, end product is lactate. The aerobic pathway produces more ATPs. Lactate pathway produces less ATP, 2 ATPs, and that around 8 ATPs. But still, there are few tissues which utilize or which perform this anaerobic respiration. It is called respiration, utilization of glucose. Which tissues? Muscle, skeletal muscle. Number 1. Number 2, RBC, mature RBC. Now my question will be to you, why this tissue prefer anaerobic respiration over aerobic respiration? Is there any condition based choice or something else? Anyone? Let's start with RBC. Shut up. Yes. RBC has no subcellular organelle. So no mitochondria, so it cannot perform aerobic glycolysis, aerobic process. Correct? So anaerobic confirmed. What about muscle? Muscle is fully supplied with blood, oxygen, everything. Why muscle will choose anaerobic glycolysis?
Because you need a continuous supply of energy for
Muscle is full of blood supply. See, I've cut a small muscle. You see how much bleeding will be there?
Yes, but in continuous exercise, energy demand becomes more higher. Energy demands becomes more higher.
Huh? Why you're not moving? Did you move? Slightly? Did you expect me to beat you? Did you expect me to beat you? Yes or no? Huh? No expectation. He has got problem with adrenaline secretion. Huh? কি পারে দেখ দেখ ওটা কি নড়ছে হ্যাঁ because you know it is not moving anything, but if suddenly anybody of you discover something is moving around your legs, what reaction you will give? Sudden jerky movement, and that is unprecedented. You don't know when it's going to happen. To survive such kind of movement, muscle cannot rely on aerobic glycolysis. It has to rely on anaerobic glycolysis. Why? Because anaerobic glycolysis is instant. It will produce less energy but instant energy. That's why, or just opposite condition to RBC, the skeletal muscle, although it is full of supply of blood, oxygen, everything, but it cannot, it prefer anaerobic glycolysis at times. If I ask you to do some exercise, what is the exercise? Come here. Go there, up and come down. Go. Run. Run. Faster. Faster, 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 faster. Go, 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 go. Faster, faster, faster. Go, 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 go. Stop, stop. What is the feeling you are having? Except the respiration rate. It is increased. What else? In your legs, what you are feeling? Cyclic cramp is there. Huh? If you do it for 50 more times, you cannot.
I can't.
So what will restrict your movement? Sit down, sit down, sit down. That is what. This is biochemistry. What will restrict your movement? What will restrict your movement that time? Lactic acid. But if the same task is given to MS Dhoni, or which cricketer you like? Siti Mandana? Virat Kohli. Talk about Siti Mandana, na? Siti Mandana. Then she can do it 100 times without even gasping for air. What is the difference? The continuous training of the muscle. More you train your muscle become more expert to deal with lactic acid. The amount of lactic acid he generated now, if you compare with that of the Mandana doing the same exercise, Mandana will produce nothing, almost negligible amount of lactic acid, because her muscles are already trained. Muscles are already trained. See, the highest level of exercise, if you ask, is nothing but swimming. Right? Have you ever seen the diet chart of a swimmer, huh? Gold medalist swimmer. He eats more than a wrestler of Haryana. He eats more than a wrestler. Why? Because he spends that energy during practice session. And he trains his muscle so that he can fly in the water. So, more you train. Now, that fat he accumulates from the diet, he spends through exercise. And the fat we accumulate from the diet, we keep it for future. We have a very good banking system. It's only inlet, no outlet. Right? So, more you train your muscle, more you get rid of lactate in faster rate. This is a net reaction of glycolysis. I need not to explain. Most of you would be knowing. What is that? Glucose plus 2 NAD. Why 2 ATP? No ATP, see? Glucose plus 2 NAD, but 2 ATP is produced. 2 NAD, 2 PI, 2 NAD, net reaction, 2 pyruvate, 2 ATP. What is happening here? A glucose is a 6 carbon containing compound. It is broken into 2-3 carbon containing compound. 2 molecules of ATP are generated by oxidation and 2 molecules of NADH are generated, which will produce further ATPs in the electron transport chain. Now, the stepwise reaction I will go, but just for every book has given this. So, what I will do? I will just discuss the important points regarding every steps. Is it okay for you, or you want me to just teach like the step 1, like the step 2, whatever you say? You want me to teach like that, like a school days, or you want me to expect that you already know something and I can just add the toppings on that? Add toppings, very good. Toppings and poppins are always good. The first reaction is a dedicated step for glycolysis. So, this step actually adds 1 molecule of phosphate from ATP to glucose at the 6th position. What is that? It's called transferase activity, hexokinase, and its isoform, one of the isoform is present in liver called glucokinase. This reaction requires 1 magnesium ion, or instead of magnesium you can use manganese. Okay, fine. So, our dication ion is required. Why this dication ion is required? Anyone? What is the role of manganese? Bolo. Hey, magnesium er role ki bolo? Hey, 2 din agei পড়েছি আমি। কি বলবি? বল। কোফ্যাক্টর, হ্যাঁ, ভেরি গুড। কি করে এটা? কি করে? কি করে? শোনো, এই যে লাস্ট বেঞ্চ, the charge over alpha and beta phosphate so that the gamma phosphate can enter well inside the enzyme molecule where it will be cleaved and transferred to the 6th carbon. One more thing, the hexokinase inside active site has got space for 6 carbons. Be it of pentose sugar, the 6th carbon position for 6th carbon will be occupied by water. So, what will happen if there is a ribose in place of glucose? What will happen? ATP hydrolysis will take place, but the phosphate will not be transferred to the carbon, rather on the water. So, there will be dissipation of energy, but no sugar phosphate will be generated because the 6th position is empty and it is occupied by water. So, pentose sugar, if pentose sugar work on it, then it will produce only ADP plus Pi, but no sugar phosphate. Alright. So, if you can remember, see the electrostatic interaction, electrostatic mechanism, what is the first point? The place where the reaction is taking place should be purely water free. What is that? Catalyzed by PHI, phosphohexose isomerase. This is an isomerase class of enzyme which converts glucose 6-phosphate to fructose 6-phosphate. It is not only converting one sugar to another sugar, but also see the structure from pyranose to furanose, pyranose ring to furanose ring. This conversion also takes place in the same step. Right? It is not only transferring one, see, sugar to another sugar, but also its isomeric form, pyranose to furanose, because fructose is more stable in furanose form and glucose is more stable in aqueous medium, and our cytosol is full of water, aqueous medium. Third step is the unidirectional. Previous two steps were both reversible. Now from here, first one was irreversible, second one was reversible, this is again irreversible. Now this step is called rate limiting step or the pacemaker step of glycolysis. Catalyzed by PFK1, phosphofructokinase 1. It is same mechanism as that of the hexokinase. Transfers another phosphate to the existing sugar phosphate and it forms fructose 1,6-bisphosphate. The term why it is not called biphosphate or diphosphate and why it is called bisphosphate, is there any query? Anyone? Everyone knows what is the difference? Good. Anyone? না জানো বলো, লজ্জা পাওয়ার কিছু নেই। থ্যাংক ইউ। অনেকেই জানে না, কিন্তু আমি কি বলি, এইটা লজ্জা করার জায়গা না, এটা শেখার জায়গা। ফার্স্ট ইয়ার মানে হচ্ছে বেসিক্স, কাকে কি বলে এইটুকুন তুমি জানবে। We are not teaching you syllable or making sentence, we are teaching you just alphabet. First year is for learning alphabet, তাই না? একদম প্রেপ ওয়ান ক্লাসের কথা মনে করে দেখো কি পড়াতো ওখানে, এ ফর অ্যাপেল, সরি এখন আবার তো অন্যভাবে পড়ায়, ফোনেটিক্সই পড়ায়। আমরা ছোটবেলায় এ ফর অ্যাপেলই পড়েছি, বি ফর ব্যাট, এখন আবার অন্যরকম বলে, তাই না? যাও একই ব্যাপার, অ্যালফাবেটে শেখে, এখন লোকের সাউন্ড শেখে, আমরা অ্যালফাবেট শিখতাম। ফাইন, ইট ইজ আ লার্নিং অ্যালফাবেট। বিস, ট্রিস, টেট্রাকিস, পেন্টাকিস এরকম যদি থাকে, আর ডাই, ট্রাই, টেট্রা, পেন্টা এরকম যদি থাকে, তফাৎটা হচ্ছে if the phosphate groups are located attached to different carbon atoms of the same molecule, they are termed as bis, tris, tetrakis, etc. If they are attached to the one phosphate to another, then they are called di, tri. যেমন adenosine diphosphate, phosphates are joined. Adenosine triphosphate, 3 phosphate one by one, correct? Whereas if there was 1 and 5, then adenosine bisphosphate, correct? So it is called fructose 1,6-bisphosphate because 2 phosphates are located at 2 carbon atoms, C1 and C6. The next step is very vital. The PFK1 is broken down, split into 2 molecules. It's a what type of enzyme? Lyase. Lyase. It cleaves a large molecule into 2 halves. What are the 2 halves? One is DHAP, another is GAP, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Again, this step requires magnesium. All steps require magnesium. glyceraldehyde 3-phosphate and dihydrogen phosphate can interconvert by Triose Phosphate Isomerase or TIM. Some people say that TIM is the most efficient, one of the most efficient enzyme discovered so far. Efficient enzyme means? Efficient means what? Higher efficiency rate means what? Suppose they are colliding 100 times with the substrate, 100 times they will produce product. That is 10% efficiency. 90%, 90% efficiency. Most of the molecules, enzymes are 40%, 30%, 60% like that. It is almost 80%. No enzyme is picture perfect. Correct? So they are interconverted. Why? Because one of them will be utilized for the next step. That is step 6. Step 6 is important because it is adding one phosphate. Adding one phosphate, and the glyceraldehyde has become glyceric acid or glycerate. That means it is an oxidation step. Aldehyde to acid. 1,3-bisphosphoglycerate. Glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. But what is produced in the side? One molecule of NADH plus H plus. So step 6. Step 7. 1,3-bisphosphoglycerate to 3-phosphoglycerate. One phosphate will be removed. Again, this is a transfer of phosphate, but this is reversible step. Only we have got only 1 and 3 so far, irreversible. Step number 1 and 3. And these produce 3-phosphoglycerate or 3-PGA and one molecule of ATP. This formation of ATP is called the first substrate level ATP formation. ATP is synthesized by two methods. One, electron transport chain. Up to ADP, there are many mechanisms. But ADP to ATP can be synthesized only by electron transport chain or by one of these reaction. One more reaction is just coming. So they are called substrate level phosphorylation, and if it takes place inside mitochondria, we call it oxidative phosphorylation. If it takes place in the cytosol, we call substrate level phosphorylation. If it takes place within mitochondria, we call now obviously the oxidative phosphorylation is more efficient because it produces more amount of this thing. But there is more similarity between these two than dissimilarity. If you can recall, the electron transport chain components are located on the membrane of mitochondria. What do you think about this enzyme? This enzyme is also bound to cell membrane. Nowhere, no enzyme can transfer electron floating in the cytosol. No. They must be bound to a place, a membrane. Why? Because membrane are lipid in nature, hydrophobic in nature, they will repel water. Once the water is present, ATP or electron cannot move further. It will dissipate its energy. The energy will be lost for nothing. So water is not a desired molecule for our survival, for the production of ATP. Right? Because ATP will not be produced. So from now on, drink less water, na? And die out of dehydration. Next is mutase. 3-PGA is converted to 2-PGA. Next step, 2-PGA to 2-phosphoenolpyruvate. Enolase, one molecule of water is removed and one double bond is introduced. It is called phosphoenolpyruvate, PEP. ATP GTP, CTP, TTP, these are all, what do you call this? এই মলিকুল কি বলা হয় বলো? হ্যাঁ, এগুলি কি বলা হয় একসময়, কি বলে এগুলিকে? বলো বলো। হাই এনার্জি কম্পাউন্ডস, তাই না? হাই এনার্জি কম্পাউন্ডস বলতে আমরা কি বুঝি? হোয়াট ডু ইউ অ্যাকচুয়ালি মিন বাই হাই এনার্জি কম্পাউন্ডস? বলো। যাদের ফসফেটটা ব্রেক হওয়া পরে তো কেজিন বলে একটা প্রোটিন আছে, তার মধ্যে ফসফেট থাকে, ফসফেট ওখানে ব্রেক করা যায়, এনার্জি পাই না তো। স্টোরে নাকি ইয়ে নাকি ব্যাংক নাকি? হ্যাঁ, এনিওয়ান who is very good in chemistry? Who is very good in chemistry? Okay, this is ATP, this is ADP. কনজুগেট বই না, যেমন এটা কি বলি অ্যাসিড, এটা কি বলি অ্যাসিড কনজুগেট, তাই না? তাহলে এই অ্যাসিডটা যদি স্ট্রং না উইক? কেন? এটা উইক বলে, স্ট্রং বলে, তাহলে এই স্টেটটা মোর স্টেবল, তাই এটা উইক অ্যাসিড। যার কনজুগেটটা, the molecule whose conjugate is more stable, most stable, it is called highest energy compound. So ADP is much more stable than ATP. That is why ATP is considered as. Same thing happens with GTP, CTP, DATP. Huh? All these are high energy compounds. Why only DATP? Hey, only ATP. DATP is there? Huh? So they are all high energy compounds. Why? Because their conjugate is more stable than the molecule itself. How much energy does it produce? Breakdown of one phosphate from ATP. on an average 7.2 kilocalorie per phosphodiester linkage or phosphate linkage, right? Phosphate bond. Phosphoenolpyruvate has got highest almost 3 times. সংখ্যাটা একটু বোঝায় আমার গন্ডগোল। 14 বা 22 একটা গলিয়ে ফেললাম, ইয়ে হচ্ছে মেমোরি ডিপ্রেস করছে, তাক ধরে নাও 22 ধরে নাও। It is the highest energy dissipating compound. So the highest energy compound is what in our system? Phosphoenolpyruvate, not ATP. Next comes creatine phosphate. Next comes all ATP, GTP, CTP, TTP. Okay? Last step, the step number 10. This is irreversible step catalyzed by pyruvate kinase. This particular enzyme require, in addition to magnesium, one potassium. Okay, this is the difference. It converts phosphoenolpyruvate to plain pyruvate and one molecule of ATP. So, this is the second substrate level phosphorylation step. Fine. Now, let us discuss the whole pathway is here. Now, let us see step number step 1 and 3. ATP requiring step. Irreversible steps. They are also called rate limiting steps. Seven and 10 ATP producing step. Okay? মনে থাকবে? তাহলে rate limiting step now we come for inhibitors. Step 1 hexokinase glucokinase. Insulin, fructose 6 phosphate, fructose 2,6 bisphosphate. এটাকে কি বলা হয়? অ্যালোস্টেরিক মডুলেটর। সিট্রেট আর গ্লুকাগন ইনহিবিট করে। তো কোনটা আছে? Glyceraldehyde 3-phosphate dehydrogenase. Iodoacetate. তারপর কোনটা আছে? Step number 7, phosphoglycerate kinase. arsenate. Number 5 কোনটা আছে 7 পরে 8 নম্বর স্টেপটা কি আছে নাম্বার 8 মিউটেজ 9 নম্বর সরি 9 নম্বরটা fluoride. Okay. 10 নম্বর ওকে এছাড়াও আছে কি আছে বলতো যদি এটিপি বেশি থাকে এটিপি এডিপি এগুলিকে বলা হয় অ্যালোস্টেটিক মডুলেটর ঠিক আছে ইফ দেয়ার ইজ মোর এনার্জি দেন ইট উইল ইনহিবিট হ্যাঁ ফাইন তাহলে এগুলি হলো ইনহিবিটরস অফ গ্লাইকোলিসিস কি কি শিখলাম আমরা হেক্সোকাইনেস সাবস্ট্রেট level of inhibition sorry product inhibition glucokinase hormonal regulation তাই তো এই কটা বাজে now step number 11 when there is situation like fight and flight or no mitochondria, the pyruvate is oxidized further to lactate. And the reaction is catalyzed by lactate dehydrogenase. And this reaction utilizes the NADH produced at step 6. Step 6 যে NADH তৈরি হয়েছিল সেই জিনিস ব্যবহার করে নিচ্ছে না so thus they are actually reducing the production of ATP by electron transport chain. Now, energetics of glycolysis. Give heading. Energetics of glycolysis. Lekho. Write down. Quickly. Leave the center part khali, vacant, which for some purpose. There is a purpose. Okay. So these 6 ATP are produced from these 2 molecules of NADH via electron transport chain, considering that no energy is lost or leaked during the process of oxidation. 1 molecule of NADH will produce 3 molecules of ATP as a whole. Yeah. PGK, phosphoglycerate kinase. Why 2 ATP here? Because from step 4 it has split into 2 halves, so twice the reaction. and Mr. Amir Khan with 2 ATP. PK? PK ho gaya. Now if we add, do we get plus 8 ATP? Okay? হয়েছে? তো লেখা হয়েছে? And in case of anaerobic glycolysis, this step will come, lactate dehydrogenase, minus 2 NADH, minus 6 ATP. So in case of aerobic glycolysis, plus 8. In case of anaerobic, plus 2. Right? For the timing, you remember this. Later on, I will come to more complicated calculation. Now you are thinking, sir is not telling, actually it is not 3, it is 2.5. Huh? 2.5 molecules of ATP, it is not 3. Everyone is telling now inside, sir doesn't know, sir is like playing like we are fool or what. We don't know that much also. I am fool. I know what you know or what you don't know. You don't know what you don't know. We know what you don't know. You don't know what you don't know. We know that teacher, we know that you know, we know what you don't know. So basically, the glycolysis is regulated by the ATP surge. If there is more ATP, glycolysis will be low. If there is less ATP, then it will be activated. Right? Control points 3. এগুলি আমি অলরেডি ডিসকাস করেছি. If you want, just go through. Already I have discussed. Those who have taken running notes, their answer is already ready. Okay, this is not required so. Okay, you don't need to remember. This will come later. This diagram will come later. This one you remember. দেখো এই যে এখানে লেখা আছে, এই যে ঠিক না, সব ঠিক আছে তো? Let's check it whether I have written correct or not. Correct, na? Okay. Any mistake is there? Tell me. See, this is the process how I make you read. And I am so clever, even if I do mistake, I will tell you, if you find a mistake, see, that is why I wanted to know that you can point out my mistake or not. How clever you are. Remember my first class, I told you the most dumbest looking teacher is at least 1,000 times more intelligent than you are. Do you agree? You? Lady? এই তুমি দাঁড়াও, হ্যাঁ, কালো কারিগান, বাংলা বোঝো? তুমি কি এটার সঙ্গে একমত যে তোমার থেকে দেখতে খুব বোকা, মানে আমাকে বোকা দেখতে লোকটাও তোমার থেকে হাজার গুণ বেশি বুদ্ধিমান? এগ্রি করো না? আমি আরো বেশি বুদ্ধিমান বলছো? হ্যাঁ, কি মনে করে তোমার থেকে কম বুদ্ধিমান? হ্যাঁ, কনফিডেন্স। কি বলে Da to hocche. Meet me after the class. Such false conception should be cleared at early stage. Fine. Now this one. This fructose 2,6-bisphosphate, this one is called an allosteric modulator for PFK1. The fructose 2,6-bisphosphate is synthesized by PFK2. It acts on fructose 6-phosphate and produces fructose 2,6-bisphosphate, not 1,6, 2,6-bisphosphate. Fructose 2,6-bisphosphate is then again brought back to fructose 6-phosphate by fructose 2,6-bisphosphatase. Now these two seemingly oppositely acting enzymes are nothing but same molecule. They are called what? Like an intelligent fellow. Darao, stand up. Intelligent to. Answer করতেই হবে, বলো। They are called একই এনজাইমের মধ্যে দুটো অপোজিট অ্যাক্টিভিটি আছে, তাকে কি বলে? দাঁড়িয়ে থাকো। bifunctional enzyme বা tandem enzyme. Bifunctional enzyme or tandem enzyme. Same enzyme has got two activity, PFK2 as well as fructose 2,6-bisphosphatase. Same molecule, same polypeptide, but two activity. If they are phosphorylated at C-terminal, they act as phosphatase. If they are not phosphorylated, they act as kinase. Same enzyme, two different activity. Depending on hormonal status, insulin promotes production of fructose 2,6-bisphosphatase. That means insulin causes generalized dephosphorylation. So insulin causes dephosphorylation of the C-terminal of PFK2. Write it down when I am telling. You are playing so smart as if you hearing once you will remember forever. So insulin causes generalized dephosphorylation. So insulin causes generalized dephosphorylation. Dephosphorylation means insulin prefers glycolysis. So insulin prefers formation of fructose 2,6-bisphosphate. So insulin prefers PFK2 activity. That means dephosphorylation of the enzyme PFK2 activates PFK2 and PFK2 produces more fructose 2,6-bisphosphate. More fructose 2,6-bisphosphate, more activity of PFK1, more and more fructose 1,6-bisphosphate is produced. That is how insulin regulates PFK1, not directly, indirectly. Sit down. Next time try to listen in the class. Don't be lost. Okay. So fructose 2,6-bisphosphate is an allosteric modulator which is synthesized by PFK2, which is broken down by fructose 2,6-bisphosphatase. Insulin stimulates production of PFK2 by dephosphorylation at C-terminal end of the molecule.